This is Microbiology, Lecture 16, Part 2. This diagram is just to remind you that the entire body is drained, the interstitial fluid, any fluid that enters into the tissues, does drain into the lymphatic vessels, and all of those lymphatic vessels eventually do lead back into the bloodstream. They enter into the fluid will enter into the blood at the right lymphatic duct or the thoracic duct. So the thoracic duct actually, the thoracic duct in fact is the entry point into the bloodstream is right here. It enters into the left subclavian vein, so it's draining tissues from most of the body into the left subclavian vein. The right lymphatic duct drains fluid on the right side into the right subclavian vein right there. Now I remind you that that tissues are constantly being bathed by fluid from the bloodstream, that the fluid that enters into the part of the fluid blood that enters into a capillary goes out into the tissues, and most of that gets pulled back in, but some of it stays inside the tissues and will flush through the tissue and enter, and I'll show it here, enter into the lymphatic. It drains into the lymphatics. You can see it shown in this way, draining into the lymphatic vessels, which means that all of our tissues are constantly being bathed by this flow, continuous flow of interstitial plasma fluid going into the interstitial tissues between the cells and then draining into the lymphatic vessels, and all of that flows back and eventually enters back into the bloodstream up here and up here at the right lymphatic duct and the thoracic duct that go into the right subclavian vein and left subclavian vein. In this way, any foreign molecule or structure that gets into us, including microorganisms, will eventually get flushed. Molecules from them or the organisms themselves will get flushed through into the lymphatic system. So one of the characteristic features of the inflammatory response is a fever, and the body's basic temperature is set by the hypothalamus, and the normal body temperature, of course, is 37 degrees. However, with a gram-negative infection and release of endotoxin from dying gram-negative organisms, endotoxin is the same, of course, as LPS or lipopolysaccharide, that will trigger macrophages, and also, of course, some macrophages will phagocytose and kill the gram-negatives, so again they get exposed to endotoxin. Endotoxin triggers macrophages to produce a cytokine called interleukin-1 or IL-1. And IL-1 is secreted out into the tissues and gets into the bloodstream and goes up through the lymphatics, goes up to the brain to the hypothalamus, which can bind IL-1 because it expresses IL-1 receptors. And the hypothalamus will be triggered to release prostaglandins that reset the basic temperature to a higher level. And that results in a higher rate of metabolism and shivering to raise temperature. The higher temperature and higher rate of metabolism overall in the body does help in response to infection, but if it rises too much, can be quite serious. Okay, now in the bloodstream, in the plasma, are a series of proteins. They are synthesized in the liver and secreted into the blood plasma, just like albumin is, or some of the clotting factors are. And in fact, it has some similarities, these proteins that I'm talking about, which together form the complement system, have some similarities to the clotting system of proteins, the clotting factors, because it does form a kind of cascade event of activation. You know, if the first clotting factor is activated, it activates others, and when those get activated, they activate more. So it's a kind of a clotting cascade as each sequential factor is activated, and you get a massive increase in these activated clotting factors. So they're present in the blood, and they can become activated in a rapid flow type of cascade. Their activation can occur via two different pathways. There is the classical pathway of activation, and there is the alternate pathway of activation of the complement system. I'm going to go through each of these, and you'll see how that works out. In the classical pathway of activation, what you get is the fact that a microorganism like a bacteria is present in the bloodstream. What you get are that the bacteria have foreign structure molecules on their surface. These are recognized as non-self antigens. And sometimes there will be antibodies in the person. Antibodies develop and they bind onto the surface. Here are two antibodies. With binding of one class type of antibody, IgG antibody, remember there are 5 different classes of antibodies. With IgG binding, you get activation of the first component of the complement system, C1. And C1 goes on to activate C2 and C4, two other proteins, complement proteins. C2 splits into 2A and 2B, and C4 splits into 4A and 4B. And these two together will activate C3. So the sequence of events so far here is C1 followed by 2 and 4 acting on 3. And C3 splits into C3A and C3B. Now C3A can trigger an inflammatory reaction that will attract phagocytic cells. And C3b carries out what's called opsonization. It will attach to a microorganism and increase the probability that the organism, the microbe, will get phagocytosed. That's what opsonization is. If a microorganism is opsonized with C3b, it's more easily phagocytosed by phagocytic cells. The alternate pathway is different. There are molecules on microorganisms that can directly activate C3. Remember, in the classical pathway we had 1, 4, 2, and then 3. being activated. Here we have C3 directly activated due to molecules on the surface of some microorganisms. And again, C3A triggers inflammatory chemotaxis. I'm going to write that out, chemotaxis. It attracts phagocytic cells to the area, which is good because they'll move into the area, they'll be brought in, and will phagocytose and hopefully kill the microorganism. It also mediates opsonization, not C3A, but C3B mediates opsonization, increasing the likelihood that the cell will get phagocytosed. Notice that in the diagram for the classical pathway and in this diagram at C3b we have this arrow going towards cytolysis. Okay, so we were discussing the complement activation of complement proteins that are found in the blood plasma, and we talked about the classical pathway and the alternate pathway. Notice that the classical pathway starts with antibody bound onto the surface of a microbe, and then you get activation of C1, 4, 2, and then C3. And the alternate pathway starts off with C3 activation directly by the bacterial molecules on the surface of bacteria. But they both converge essentially on, they either start or converge onto C3, and everything else after that is the same in the two pathways, the classical pathway and the alternate pathway of complement activation are identical after that. C3 splits into, when it's activated, splits into C3A and C3B. And C3B activates C5 to split it into C5A and C5B. And C5B with C6, 7, 8, and 9 form the membrane attack complex that can insert itself into the bacterial plasma membrane and cause rapid leaking out of the contents of the cell and therefore death of the bacterial cell. I should point out to you that both C3A and C5A have chemotactic abilities. They cause inflammation by increasing the attraction of phagocytic cells to site, but they can, both of them have chemotactic ability. They can also act directly on mast cells. Mast cells are basophils that have left the circulation and entered into tissues and are filled with granules. And when these two, either one of these two, act on mast cell, they causes degranulation and release of histamine, which is in the granules. And histamine itself causes an inflammatory response in that it causes an increased vascular permeability. And therefore localized edema, also vasodilation and increased vascular permeability. And so when you look at the effects of complement activation, C3b does cause opsonization, resulting in an increase in phagocytosis of bacteria. Both C3a and C5a have chemotactic activity. They can attract phagocytes from the phagocytic cells from the circulation into the area. C5a has more activity than C3a, and some sources that you might look at would in fact say that they would leave out C3a, but they both do have activity, although C5a appears to have more. Both of them can, either one can cause degranulation of mast cells, which are basophils that have moved into the tissue, and therefore cause the release of histamine, and histamine causes vasodilation, so there's an increase in flow of blood into the area and increased vascular permeability, so more fluid leaks out, you get edema, and also it's easier for cells to move out of that leakier capillary, local capillary. And of course C5b, 6, 7, 8, and 9 together can form the MAC or membrane attack complex. And so, you know, complement activation is an important thing. These photos show, these electron micrographs show on this side a bacteria that has had a membrane attack complex punch a hole in its surface. Here you see the effects of C3A and C5A. I've added C3A here to this diagram. The two, either one can attract neutrophils or macrophages, phagocytic cells, in other words, to a site. And here you see the action of either C5A or C3A binding to a mast cell and triggering degranulation of the histamine, release of the granules containing histamine. And so some bacteria can actually avoid or escape from the effects, evade the effects of complement. Some have capsules. Capsules not only are good at reducing phagocytosis, but they can prevent complement activation. Some have surface molecules that can prevent MAC formation, membrane attack complex formation, and some organisms have enzymes that can break down or digest C5A, thus reducing the effects of complement. There are many cytokines that are produced in the immune response. There's a huge number of them. Some go by the nomenclature naming interleukin. We already discussed interleukin 1, which is produced by macrophages exposed to endotoxin or LPS from gram-negative bacteria, and that gets released in the bloodstream and can trigger a response in the hypothalamus to raise the body temperature. But there are many others besides, there are many number, 1, 2, 3, 4, 5 on, of interleukins. These are protein molecules that act as signals between white cells. Some don't go by that interleukin naming. Here, example, interferon. Interferons are molecules, cytokine molecules that trigger production of antiviral proteins. That is at least alpha and beta interferons. Gamma interferon is a cytokine that can activate macrophages and neutrophils to be even more phagocytic, and also to produce some more inflammatory molecules and be more active. And here's a diagram that shows, you know, the effect of interferon produced by alpha or beta interferon produced by one cell, triggering production in a neighboring cell of antiviral proteins. I'm not going to go into the details of this. It's for your own interest if you want. So we have spoken now about when we discuss mechanisms of pathogenicity. As I said at the beginning of this lecture, lecture 16, we said that in order for an organism to gain a foothold and successfully infect a person, it not only needed a portal of entry, but also had to penetrate or evade host defenses, cause damage, and have a portal of exit so it could go on and infect another person or individual. And here we've talked about some of the aspects of these as part of the innate immune defense mechanisms, the innate immune system, the first line of defense and the second line of defense. Not everything in the second line of defense is listed here, but you know, it's an important thing that if it's going to successfully infect you, it has to get around these defenses, and the defenses are quite active. You are constantly, in fact, having pathogens on you or in you, and they don't succeed because of the innate immune system, and you don't even realize it. You have very little, if any, symptoms at all. Okay, so that's the end of Lecture 16, Part 2.